Good day, gentlemen. Today is March the 26th, and today we're going to talk about section 14.4 in the chapter about Mendelian genetics, and that is that many human traits do follow Mendelian patterns of inheritance. However, studying human beings can be very difficult in terms of inheritance patterns because the generation time is so long. All right, oftentimes it's 20, 30, even 40 years between generations here, and parents only produce a few kids, and so it can be very difficult to trace a gene or an allele through a population. Now, one way that this can be done and analyzed is by using what we call a pedigree. Now, a pedigree is a chart essentially that labels each individual in a family and essentially writes down what their genotype is. Okay, so for example, if grandma here is homozygous recessive, but grandpa shows the dominant trait, if any of the children show the recessive trait, that means that grandpa had to be carrying the recessive allele, right? Because they get one allele from each grandparent. Now, and so you can use clues in terms of the phenotype of each individual in a family in order to figure out what the inheritance patterns actually are. Okay, the pedigree itself shows matings, it shows offspring, and then here's another mating, and then the offspring here. The two traits shown here, widow's peak, you can see the little peak in her hair here, whereas her sister does not have that peak. The other one is a taste test. There's a certain chemical called PTC that some people can taste and some people cannot. In the case on the right, it's a recessive trait. The widow's peak is a dominant trait, just for kicks and giggles. Okay, so let's start with recessively inherited disorders. Okay, many genetic disorders are inherited in a recessive fashion. An example of a recessive gene that is passed on is the one that causes albinism, making a child, a child, an albino. Now, recessive disorders can only show up in individuals that are homozygous for the recessive allele. So here's the young lady on the right-hand side has two recessive alleles for albinism. All right, the other three boxes in the Punnett square are either homozygous dominant, which shows the normal phenotype, or heterozygous, which shows, again, the dominant phenotype. But these two individuals, all right, are both what we call carriers, because even though they show the dominant phenotype, they carry the recessive allele, all right, and so they are called carriers. And again, albinism is an example of a trait that can be carried recessively in somebody who shows the dominant phenotype. Now, usually under normal circumstances, recessive alleles and the diseases that they cause are very rare because it's very unlikely that two carriers will actually meet and produce an offspring. However, when inbreeding occurs, all right, and this can occur in humans and in other animals, you then have a much higher chance that the offspring produced will have the recessive phenotype, because it's much more likely that those two recessive alleles will come together during the fertilization event. Okay, this is one of the reasons why most societies and cultures have taboos or cultural laws against inbreeding, all right, marriage between close relatives. Another recessively inherited disease is cystic fibrosis. It is a disease that causes the buildup of mucus in the lungs because of some chemical aspects. There's an enzyme that doesn't allow chlorine ions to move in and out, and mucus builds up in all of the organ systems. But the disease itself isn't important right now. It is a recessive allele, and you can read more about this in your textbook. Sickle cell anemia is another fascinating recessively inherited disease in that although the disease itself is terrible, it turns out that carriers may actually have an advantage, which might be why the gene has been propagated. So sickle cell anemia affects about 400, 1 out of 400 African Americans. And it's caused by a simple switch in one amino acid in the protein that forms the hemoglobin in red blood cells. Now, in a person who is homozygous for this, they will actually have hemoglobin that is abnormal. And that abnormal protein causes the red blood cells to take on a sickled shape, looks almost like a crescent moon. And this can cause weakness, pain in the joints, difficulty, fatigue, that sort of thing. Now, heterozygotes are said they have the trait, but they're typically pretty healthy. They don't really show the sickle-celled traits. And about 1 out of 10 African Americans actually has the trait, is a carrier for this. But here's the interesting thing. It turns out that heterozygotes are less susceptible to malaria. All right. And so this may explain why evolutionarily this recessive allele, even though it's so damaging and disabling, has been passed on for generation after generation, because it does confer some immunity to malaria on the folks who actually carry the allele. This is just a brief explanation of how this works. You've got, here's a person who is homozygous recessive, 2 sickle-celled alleles. The proteins that they produce for their hemoglobins cause the red blood cell to be sickled in terms of shape. Here is a person who is a heterozygote, 1 sickle-celled allele, 1 normal allele, and the proteins that they produce allow some of the red blood cells to be sickle shaped, but some to be normal, and therefore they have, they are able to carry enough oxygen to their cells and all of that kind of stuff. Now, diseases are not just inherited, or conditions are not just inherited dominantly. Dwarfism is an example of a dominantly inherited disorder. The allele that causes dwarfism is a dominant allele, and therefore a dwarf person, somebody with achondroplasia, even if they are a carrier, all right, because they've got the dominant allele, but then a recessive allele, they can still produce dwarf children. Now, interestingly enough, they can also produce normal children of normal stature, because if they meet or breed or marry somebody who is homozygous recessive, then there's 50-50 chance that their children will be either dwarfs or of normal height. Another aspect of this is timing of when a disease actually begins affecting the individual who carries it. Huntington's disease is a good example of this. This is the disease that causes a breakdown of the nervous system, and people have absolutely no symptoms or signs until they get into their 30s, 40s, and 50s. And by that point, they've probably already gotten married, started a family, and that sort of thing. And, of course, already passed on the gene. And so you don't know that you are a carrier of this disease or even affected by this disease until well into adulthood and even old age. Certain diseases are affected by, certain genetic diseases are also affected by the environment around them. And so no matter what your genotype, your lifestyle and your environment has a huge effect on what you look like, what your phenotype is going to be. Now, there are ways to go about getting tested genetically. A genetic counselor can provide lots of information through pedigrees, family histories, and even karyotypes and blood tests. Oftentimes, these genetic counselors will use Mendelian genetics and probability rules in order to figure out what's going on and what are the chances that a couple will produce a child with a certain condition. All right, I'm not going to go through all of this, but if both members of a couple had a sibling with a recessively inherited illness, then that means that both of their parents, in other words, all four grandparents, were carriers. And therefore, each of the children has a 2 in 3 chance of being a carrier. The fourth, of course, option is the person who actually, the brother who inherited that illness. So there are only three boxes left on the Punnett square, and 2 out of those 3 are carriers. So if both of them are carriers, then they have a 25% chance of producing a child who has the illness. There are tests for identifying carriers. These are genetic tests that can be done through a blood test or other things. One of the ways that we can also examine the probabilities are by doing exams on fetuses inside their mother's womb. Amniocentesis is when they remove the liquid from inside the amniotic sac. So you can see the syringe going into the mother's abdomen, right through the wall of the uterus and the placenta, or the uterus, actually, withdrawing that fluid. Skin cells from the baby inside that fluid can be tested then and looked at and formed a karyotype. In chorionic villus sampling, a sample of tissue is removed directly from the chorion, which is the lining of the placenta, and that can be tested genetically as well. And then finally, there are certain tests that are performed and can be performed on newborns to determine what their genetic predisposition is. There's one for PKU, or phenylketonuria. This is a disease that is, I believe it's a faulty fat, a depositing of fats on nervous systems, and it. But I don't honestly remember this. But it affects 1 out of every 10 to 15,000 births in America. All right, but it is a test that's done on babies right after their births. All right, boys and girls, that brings us to the end of our slideshow for section 14.4. So, good luck.